Yeah, but maybe next time. So I will give you an overview about our research, and I will give you, tell you something about our main interest, which is the differences between commensalism and pathogenicity of Candida albicans, and I will start with this slide, which shows you where we are. Here we are in Europe, and this is Germany. Jena is here, and this is the city, and these are the two buildings of our institute. My office is in this building here. We are a member of the Leibniz Association, which is a bit similar to Max Planck Society, but our association is having the idea of theoria cum praxi. So we always do basic research with applied ideas at our institute, which has two foci, natural product research and infection biology. And here there's a focus on fungi. We have 7 departments, research groups, junior research groups, and a graduate school and National Reference Center. So this is the Beutenberg campus where we are located. There are Max Planck Institutes, Fraunhofer Institutes, industry, university, 2 more Leibniz Institutes. So it's very focused here. All right, let me start with the general topic with environmental fungi, with numbers of fungi existing in the world. So there's an estimation that there are about 3 to 5 million fungal species, so a huge number of fungal species. Many of these are actually living in harmony with hosts. In particular, higher plants are almost all associated with fungi, and these are the mycorrhizae. But you all know lichen, for example, as a symbiotic interaction of fungi and algae. So this is actually symbiosis. But then you have pathogenic fungi, and in particular, plant pathogenic fungi are very common, like these species here, Magna porta griseae, Pyronophora, Ustilago, Fusarium, Botrytis, Colitrichum. These are all plant pathogenic fungi, and in fact, plants, the infection agents of plants are particularly fungi, and they cause a huge loss in agriculture. So it's estimated that there are about 125 million tons of destroyed plants due to fungal infections, with the top 5 food crops: rice, wheat, maize, potatoes, and soybeans every year. And it's estimated that there's about a loss of about $60 billion in agriculture due to fungi, just to rice, wheat, and maize alone. So it's a huge loss. And this is due to approximately 8,000 plant pathogenic fungi. So considering how many fungal species exist, this is a little number, but if you look at human pathogenic fungi, then there are only about 150 species which can actually cause disease in humans. So that's less than 0.04% of all fungi existing, which are potentially human pathogens. Nevertheless, these few species infect billions of people every year. The majority of these infections are superficial infections of skin, mucosal surfaces, and nail. It's estimated that about 25% of the world population is actually infected with dermatophytes. This is skin and nails. But also 3/4 of all healthy women experience at least one vaginal yeast infection in their lifetime. And in HIV-infected patients, more than 90% of these get oral Candida infections. So it's very, very common. But there are also life-threatening fungal infections. And in fact, fungi kill at least as many people as tuberculosis or malaria. Recent numbers suggest that there are three times more fungal infections, more dead people due to fungal infections as compared to malaria. So it's a huge number and it's underestimated. Here are the major killers, the hidden killers: Aspergillus, Cryptococcus, Candida, Mucor species or Rhizopus species, and Pneumocystis. These exist worldwide. And you can see the estimated number of life-threatening infections per year. So that's huge. The problem is also that these infections are, one of the problems, these infections are in hospitals. So these are the nosocomial infections, and particular Candida is important here because Candida species are the most frequent nosocomial fungal bloodstream isolates. And the mortality of severe sepsis caused by Candida is greater than 50%, so that's higher than most bacteria. Estimated 400,000 life-threatening Candida infections per year, and it's probably more than these. Candida species are shown here. That's most of them, the top 4 are shown here. Candida albicans, Candida tropicalis, Candida parapsilosis, and Candida glabrata. And these 3 here belong to the so-called CUG clade. That is a clade where the CUG code is actually read as a serine, not as a leucine, as the normal general genetic code. Candida glabrata, which belongs to the top 4, is actually more closely related to Saccharomyces cerevisiae as compared to these 3 others. So they all belong to the genus Candida, but they are not really related. And then we have moderate pathogenic fungi like Candida duplinensis, and then the rest I would name as non-pathogenic. So let me talk about the most common Candida species, Candida albicans. This fungus is associated with animal and humans. It's an opportunistic pathogen as it can only cause disease under certain circumstances, and it's responsible for more than 50% of all Candida infections. Candida albicans is polymorphic as it can grow in different morphological forms, like a yeast form, a pseudohyphal form, and a true hyphal form. And I call it the most common human fungal pathogens because the majority of the population is carrier of this fungus. I would guess it's even up to 100%. So potentially we are all carrier of this fungus. But that means we have 2 different lifestyles. We have the commensal lifestyle and we have the pathogenic lifestyle. So as a commensal, Candida albicans is on mucosal surfaces, oral cavity, vagina, the gut, probably most in the gut. Then as a pathogen, the fungus can very frequently, I just mentioned vaginal infections and oral infection of HIV patients, very frequently cause mucosal infections, surface infections. Also skin can be infected. But then the fungus can potentially invade through the epithelial layers and can reach the bloodstream. Or nosocomial infections, iatrogenic factors. Surgery is a risk factor. So is drug treatment where the epithelial barrier is damaged. And some fungi, some Candida albicans cells can spread from biofilms on catheters in the hospital. So that's clearly a hospital-associated situation. And these bloodstream infections, they are life-threatening. From the blood, the fungus can infect almost all organs of the human being, and as I said, this is life-threatening. So in principle, we have 3 different stages. We have adhesion, invasion, and damage, from commensalism to pathogenicity. And these 3 stages can be further divided to further sub-stages, which is ad hoc adhesion, induced adhesion. Invasion can be induced endocytosis or active penetration. And damage requires hyphal elongation and destructive activities. And I will speak about each of these steps. I will also speak about nutrient acquisition because you can imagine that a fungus, when it lives on mucosal surfaces, in particular in the gut, and it's now invading into tissue, it has to change its strategies to get nutrients. And also sooner or later, the fungus will meet immune cells and has to develop or has to use immune evasion strategies. And finally, I will speak about what I just mentioned, the translocation from the epithelial cell, from the epithelial surface into the bloodstream. And that is called translocation. Right, let's start with adhesion of Candida albicans to epithelial cells. This is shown here. A graph shows the adhesion, and you can see it is a dynamic process. Adhesion properties increase with time. So why is this the case? This little video will show you yeast cells in liquid on a plastic surface. They start floating around in the liquid, but then they stop floating because they produce hyphae. And the reason for this attachment is because during the yeast to hyphal transition, there are hyphal-associated genes expressed, ALS3 and HWP1 in particular. And these proteins, corresponding proteins, are on the surface of these hyphae, as shown here. And they are adhesins. That is not only happening on plastic surfaces, but also on epithelial cells in the lab, but also in patients. You can see this epithelial cell from the oral cavity. You can see the hyphae being attached to this epithelial cell. So what's the evidence that this is really true? We all produce, as molecular mycologists, mutants. And if you have an HWP1 mutant lacking HWP1 or ALS3, you have about 70% or 60% reduction in adhesion. And if you knock out RAS1, which is one of the key regulators of the yeast to hyphal transition, shown here, you have almost no adhesion left. So that clearly shows that these proteins on the surface are mediating attachment to surfaces, epithelial surface in particular. Next, invasion of Candida albicans into epithelial cells. As I mentioned, there are two different routes of Candida albicans invasion into epithelial cells. One is induced endocytosis and one is active penetration. Induced endocytosis is an entirely host-driven process. You don't actually need fungal viability, but you need hyphae. And the reason for this that there is an invasion which triggers the uptake of Candida albicans into epithelial cells, and that is actually ALS3. So ALS3 triggers its own uptake. You can see this membrane ruffling here, and you can see that this hyphal cell is almost sucked up by the epithelial cells. So ALS3 is not only an adhesin, but it's also an invasion. Then we have active penetration, and that is, in contrast to induced endocytosis, a fungal-driven process. You don't need any viable host cells. You can kill the host cell and you still have this, but you need hyphae. And depending on the cell type, you have, in this case, oral epithelial cells, minor contribution of induced endocytosis, about 25%, and the dominant route is active penetration. This way of getting into host cells, into epithelial cells, that's used by bacteria. And this is used by parasites and plant pathogenic fungi. So here's one example of Magna portagrisii. Here's the spore. It has produced a germ tube, and then the fungus produces a specific structure which is called an appressorium. And with this structure, the fungus is pushing fungal elements into the host tissue. And that is called a hostorium. And this is surrounded by a membrane. And this is very important for the communication of the fungus with the host, an extra hostorial membrane. And we propose that we have something similar in the case of Candida albicans. Here we have induced endocytosis, again, active penetration. And if you look at the tissue underneath, you can see here, more closely here, that there's a membrane surrounding this invading hyphae. And if you use trans-electron microscopy, it's even clearer. So this invading hyphae is surrounded by a membrane, shown here, right? So we call this the invasion pocket of Candida albicans hyphae. So next, as I told you, nutrient acquisition is important. And one of the main nutrients required during invasion or required by all microbes is iron. So the fungus has to change its strategies to get iron from the host when it's invading. And we observed that invading hyphae are actually surrounded by ferritin. And we were wondering which factor might be important for this binding, and we found that this was actually ALS3. So ALS3 mutant is unable to bind ferritin. The revertant, where we put back the gene into the mutant, is still able, or again able, to bind ferritin to its surface. And ferritin is the main iron storage protein of epithelial cells. Loads of iron in ferritin. And the proposal here is like this. You have ferritin binding to ALS3. Then you need acidification to release the iron from ferritin, and then you have the reductive uptake system of the fungus, which enables the fungus to get iron into the host, into its own cell. So that is iron acquisition, one example of iron acquisition during epithelial infection. The other one is zinc acquisition. Zinc is as important as iron. So microbes as host cells need zinc, and the fungus has to develop systems or has to use systems to get zinc from the host while it is invading. And here we observed a very similar situation. We have the hyphae invading into the host cells, into the epithelial cells, and you can see that those parts which have invaded, here we have accumulation of zinc on this invasive hyphae. And the system here is different from the system which I just explained to you in the case of ferritin. Here we have a zincophore system. We call this zincophore because the system is similar to siderophores, which are required for getting iron from the environment. In this case, we have a factor which is called PRA1, which is the siderophore, which is secreted. It grabs zinc from the host, returning to the fungal hyphae, and it delivers zinc to ZRT1. And ZRT1 is both a receptor of PRA1 and a zinc transporter. So that is a zincophore system, a very sophisticated, nice system to get zinc from the host. Right, so, but how does this fit together? On one hand, I have told you that the invading hyphae is surrounded by membrane, but then I told you that it's accumulation of ferritin on the surface and accumulation of zinc on the surface. While surrounded by membrane, that does not fit. How does the fungus get access to the host cell cytoplasm? And here damage of epithelium comes into play. And we propose that here at the fungal tip, there is where the fungus can actually cause damage. So we were looking for mutants which are less able to cause damage of epithelial cells. And there was one mutant which lacks EED1. This mutant has normal abilities to attach to oral cells, normal abilities to invade oral cells, but there's almost no damage anymore. So EED1 is essential for cell damage, but not adhesion or invasion. So why is this the case? Let's go back to the same movie which I have showed you at the beginning. Yeast cells in liquid on a plastic surface. They're floating around and they stop floating because they produce hyphae. Now the EED1 mutant is lacking EED1. Similar situation, also not as strongly, but they produce hyphae. However, after 3 hours, about 3 hours, something is changing. There's actually no hyphal extension anymore. And in contrast, the mutant switches back to yeast growth, and they are not attached anymore because it's yeast cells now. So, in summary, the wild type is producing hyphae, which adhere properly. The EED1 mutant is producing hyphae at the beginning, but then switches back to yeast growth and continues to grow as yeast cells. And this has a dramatic consequence during the interaction with epithelial cells. This is the wild type after 1 hour. I've shown you this picture a number of times now. Membrane ruffling, uptake of hyphae. That's the EED1 mutant, the same uptake of these hyphae. But then after 24 hours, the wild type looks like this, and the EED1 mutant looks like this. So these EED1 cells are trapped within epithelial cells because they can only grow of yeast cells, and obviously these yeast cells are unable to evade from the epithelial cells. So very interesting phenomenon. So summary number 1, Candida albicans hyphae formation and elongation is essential for epithelial damage. Right, next comes ECE1 into play. EC1 is a hyphal associated genes. It stands for extent of cell elongation number 1. One of the first hyphae associated genes identified in 1993. It belongs to the core fundamentation response genes, only 7 genes which are expressed under almost all conditions when hyphae are produced. And this gene is extremely high expressed during invasion, up to 10,000-fold expression during invasion. And we can also show that with a GFP reporter, which has been put behind the EC1 promoter, and you can see nicely that this gene is expressed during hyphae formation. So from during invasion, there's a high expression of EC1. So what's the role of EC1? It has not been elucidated for many, many years, and of course we made a mutant to identify what's going on here. And the mutant is completely normal to produce hyphae. Adhesion properties are the same, invasion properties are the same, but it cannot produce any damage anymore. That was surprising. So summary number 2, Candida albicans hyphae formation per se does not cause epithelial damage. You need something else. And that seems to be EC1, sorry. EC1, and if you look more closely at the corresponding protein, it's actually a polyprotein. A polyprotein consisting of 8 different peptides, which are divided by lysine arginine sites, Kr, and lysine arginine sites are well known to be processing sites of the KEX2 protease, which is located in the Golgi. So here are the Kr sites, and we actually could show using a recombinant EC1 polyprotein that when using KEX2, it's digested into peptides. And we could confirm by LCMS that all these peptides exist after the digest of KEX2, and we could also show that as an additional digest of KEX1 between the lysine arginine sites. So these peptides actually end with a K. Right, so that's the situation, and then we were wondering what the different peptides actually are doing, and we found out that one of these peptides, peptide number 3, is causing damage. So if you use synthetic versions of this peptide alone can cause epithelial damage. And we actually found, when you look more closely to this peptide, that it is similar to melitine. The melitine is the B venom, a very potent cytolysin. And the similarity is shared is that both are amphipartic, hydrophobic N terminus, 2 alpha helices, and a hydrophilic C terminus. And what do? They produce pores. And that's shown with this experiment, a nanospot array, where you have an array with little holes in it, and you can put a fluorescent dye into these holes, and then you cover it with a membrane. And then you investigate what your agent of interest is doing, in this particular adding peptide number 3, and this peptide causes pores, and the dye is released. And we can nicely quantify this. And this has been done by Thomas Gutzmann in Borstel here in Germany. And he could also show that depending on the membrane, on the lipid composition of the membrane, whether it contains phosphatidylserine or not, you have very high damage, or you have moderate damage or low damage. So with other words, we have discovered a toxin, the first Candida albicans toxin, which we named candidialysin. This is actually the first peptide toxin identified in human pathogenic fungus. And we called it the missing link between hyphae formation and host cell damage, because it was known for a long time that hyphae formation can cause damage. Right, so back to the invasion pocket now. Putting things together, we proposed that these invading hyphae in the hyphae pocket, they produce damage here. And now we used an antibody which had been produced by the company QBQ, and we could nicely show that candidialysin actually accumulates at the hyphae tip. And we also have evidence that damage occurs because, in collaboration with a group of Sergio Greenstein, they could show that ALG2, which is a repair protein, is found at the hyphae tip. So here, the epithelial cells have active repair, clear evidence that damage occurs at the hyphae tip. So nicely fits together. Right, summary number 3. The hyphae-associated peptide toxin candidialysin is essential for epithelial damage. Now, translocation of Candida albicans through intestinal epithelial layers. I told you that this translocation is required for bloodstream infections. So the Candida albicans, as in the native route, as I call it, is invading through epithelial cells and can reach the bloodstream. And this is predominantly from the gut, which is the main source of disseminated Candida albicans infections in the clinics. So we investigated this by transwell assays. This is such a transwell assay where with epithelial cells on a membrane with pores, and we measure three different things. One is epithelial barrier integrity measured by TEA, shunts, epithelial electrical resistance. So that's measuring the integrity of this layer here. Then we can measure LDH release. That is indicative of cellular damage. LDH stands for lactate dehydrogenase. And this is a cytoplasmic protein of epithelial cells. And if that's released, we know epithelial cells are damaged. And finally, we can measure the direct translocation of fungal cells. And we measure CFUs, colony-forming units. And each colony represents one cell which has managed to translocate through this layer. So these are the three readouts. And of course, we also did controls using the EC1 mutant. Adhesion is the same, invasion is the same, hyphae formation is the same. You can see here hyphae formation of the EC1 mutant. But you see integrity is clearly not that much damage as the wild type, so clearly more intact. Epithelial layer, epithelial damage, very clear difference. EC1 mutant almost unable to cause epithelial damage. But now importantly, this also is associated with reduced translocation. So these two things mediate translocation. And our model is shown here. We have transcellular invasions through epithelial cells associated with damage, necrosis, and that is the main driver of translocation. We also have a bit remaining, about maybe 25%, and that is potentially paracellular invasion between cells. We can exclude apoptosis because we have tested this. So we have the situation that we have candidialysin as a classical virulence factor. A microbe is having a gene A, which produces virulence factor A, and that's causing damage. This is a classical virulence factor. However, the question also arises whether the host is helplessly exposed to aggressive Candida albicans hyphae. And here is the so-called danger response pathway, which has been discovered by the group of Julian Naglick in London. And he could show that depending on a certain threshold of hyphae on the surface, the epithelial cells can sense that there's a danger, and then it can react via P38, CFOS. There's production of cytokine inflammatory mediators, which cause attraction of phagocytes, which actually then cause clearance of the fungus. So this is clearing of the fungus. The question was, which is the hyphal moiety, or is it damage, which is actually recognized by the epithelial cells? And we know now that it's candidialysin. Candidialysin is sensed and it's triggering the danger response pathway, which causes attraction of phagocytes, and that causes clearance of Candida on the surface. With other words, summary number 4, candidialysin induces a protective immune response. So, next interaction with Candida albicans is macrophages. I told you that when the fungus is invading, sooner or later, the fungus has to face the immune system, in particular phagocytes, including macrophages. Now, that's another movie here. These are phagocytes, macrophages, and yeast cells of Candida albicans. If you look at this phagocyte here and these yeast cells here, which are phagocytes by these macrophages, phagocytosing very efficiently, but then it's exploding because the fungus is able to produce hyphae within macrophages, and that is an escape route, but also killing mechanism. And there were a number of studies which showed that there were 3 proposed mechanisms. One is pyroptosis, so that's a controlled induced cell death associated with inflammation, physical forces, and glucose consumption. So we were, of course, interested whether candidialysin as a toxin is also contributing to this, contributing to escape from macrophages. Controls, percent of phagocytosis, the same, hyphen length, the same, hyphen escape, the same. So the escape per se does not require candidialysin. However, the damage is reduced. So there's clearly significantly reduced damage. Not as much as with epithelial cells, but the EC1 mutant clearly causes less cell damage. And this is caspase-1, ASC, and NLRP3 independent. And this is indicative that it's pyroptosis independent. It's also apoptosis independent. With other words, there's a candidialysin dependent pyroptosis independent macrophage damage, and that was new. But so Candida albicans kills macrophages, but before the macrophages die, actually the fungus cries for help because it's producing. So not the fungus, the macrophages cry for help because the macrophages induce IL-1 beta, and that is far more in the wild type as compared to the EC1 mutant. So the EC1 mutant is less able to produce IL-1 beta. And that's also clearly shown with the synthetic peptides. So candidialysin, peptide 3, increasing concentrations, more IL-1 beta production. And this is caspase-1, ASC, and NLRP3 dependent. So there's a candidialysin dependent inflammasome activation, which is not pyroptosis. So we have dual functions of Candida albicans. We have, in this case, with the macrophages, we have candidialysin-mediated inflammasome activation, but on the other hand, we have also candidialysin-mediated membrane lysis and host cell death. Two different things, very different. Host damage and antifungal host defense. So this is published here. We have the dual functions, but we not only have the dual functions here, we also have knowledge about the work from Sarah Gaffen, who showed that candidialysin is actually regulating a protective immune response on oral epithelial cells. Rebecca Drummond could show that there's a similar situation in the brain where you need candidialysin for the attraction of protective neutrophils. And then we clearly could show that candidialysin is a classical virulence factor and it's mediating translocation through epithelial damage. So dual functions of candidialysin. So the situation is as follows: dual function genes, virulence and avirulence. So in this case, microbe has a gene A which is encoding a virulence factor A causing damage, classical virulence factor. Then the microbe in the gene A encoding a factor A, now it's a factor A, which is causing damage. And that might be sensed, or the factor might be recognized by the host. And that in turn causes a protective immune response. And now the virulence factor is an avirulence factor because it causes its own cell death, clearance. So virulence and avirulence. And candidialysin is a clear example for this. Candidialysin has dual functions as a virulence factor and as an avirulence factor. And it's important to stress that this is the result of co-evolution. So you can imagine that if the fungus has produced a toxin to damage the host, maybe to get nutrients, but the host has learned to recognize this damage and to recognize this factor to counteract. A clear example of co-evolution. So summary 5, candidialysin is a dual function factor causing either virulence or avirulence. Right, and the story goes on because there's a bit similar situation with PRA1, the zinc for which I told you at the beginning. That mediates immune evasion via binding of factor H, so that's immune evasion, but it is also recognized by CR3, and that's causing phagocyte attraction. So a similar situation. On one hand, getting zinc, immune evasion with binding factor H, but at the same time, binding to CR3 causing phagocyte attraction. There's another factor, which is cell1, which has a similar situation. So we have more examples of virulence and avirulence, or dual function genes responsible for virulence and avirulence. Okay, right, so the situation is as follows. we have virulence and damage on one side and avirulence and immune response on the other side, with the example of candidiasis. If candidiasis causes too much damage, then we have disease. But if it is recognized by the host, we have immune responses, and that's clear, and maybe a shift back to commensalism. If that's too much, that depends on the tissue and the type and stage of infection. If that's too much of an immune response, then you might have immune pathology, which is disease as well. And that's actually the case in vaginal infection. Because in vaginal infection, you have attraction of neutrophils, but these neutrophils are detrimental. They are part of the immune pathology. Okay, finally, more damage screening, more screening of mutants which are less able to damage epithelial cells. And here's one run, and there's one mutant here which is clearly reduced in its ability to cause damage. And that's the mutant lacking DUR31, effective in damage of epithelial cells. This mutant has normal abilities to attach to epithelial cells and invasion, but it has reduced hyphae growth. Here's the wild type, hyphae, DUR31, no hyphae under these conditions. What is the reason for this? The reason is that DUR31 is a polyamine transporter, which mediates filamentation and thus virulence. It mediates these polyamines like spermidine, which is transported into the fungal cell. Then you have survival and growth, but also ammonium production, CO2 production. That is transported to the outside of the cell. That's causing alkalinization, and alkalinization induces filamentation. Right, so we have this factor mediating filamentation and thus virulence, but we also notice that DUR31 is also transporting histatin 5. Yes, histatin 5 fits labeled. There's less accumulation of this fits labeled histatin 5 in the fungal cell. It's quantified here, so clear reduction of uptake of histatin 5. And histatin 5 is an oral antimicrobial peptide. That means it's detrimental for the fungus. So on one hand, the fungus can take up spermidine, causing filamentation and virulence, but also it is taking up histatin 5, and that's causing cell death of the fungus. So DUR31 is clearly also a virulence and an avirulence gene. So here we can extend our model, not only that there's an avirulence function by sensing and recognition, but also by exploiting this factor here, for example, in this case, by transporting an antimicrobial peptide into the microbe. Right, finally, my final summary, Candida albicans from commensalism to pathogenicity. You have the commensal situation, you need commensal factor for this. Then you have to switch to pathogenicity, you need virulence factors for this, ALS3, PRA1, ED1, EC1, DUR31. But then you also have avirulence factors, which potentially causes a shift back to commensal growth, and that's including PRA1, EC1, DUR31, and cell1. And some of these are both virulence factors and avirulence factors, dual function factors. So I would like to stress, never forget, if you work with things like this, nothing in biology makes sense except in the light of evolution. Don't forget this sentence. Now I would like to thank the people which were involved. My department here, many people were involved here, in particular the EC1 team, as shown here. And I would like to stress that all the EC1 work, all the candidizing work is always done in collaboration with Julian Naglick at the King's College in London. We do have a global EC1 team in several places of the world. We have further collaboration partners. And finally, I would like to thank the funding agencies, which are shown here. And I would like to thank all the collaboration partners and the funding agencies. And I would like to thank you for your attention. Thank you very much.
Thank you, Bernhard. It's been a pleasure to hear how much knowledge we have gained about the Candida infections in the last years. And so for the participants, you can write questions in the chat or either if you want to ask directly, write that in the chat so I can allow you to answer. But I have myself a few questions, but I think one of the most interesting things is that the Candida lysine is also transcribed and traduced along with other peptides. So have you advanced in the role of the other peptides?
Very good question, of course. Yes, they are very interesting. And I'm happy to say that I just received a grant from the German Research Association to investigate this.
Oh, right.
Because this is non-Candida lysine EC1 peptides.
Mm-hmm.
And we propose that they have several functions. One is the folding and processing of the toxin. And we have clear evidence for this. If we just remove single peptides from the polyprotein, then we don't have proper folding anymore, which is indicated by reduced growth, reduced hyphae formation. That was a bit unexpected because we did not imagine that each of these peptides would have such an importance. But we also know from sequences from our collaboration partner at the Pasteur Institute, Christophe Enfer, that these other peptides are extremely conserved. So they must have very particular functions. So folding is one, and we believe that the other one is protection, that as it is with almost all toxins have to be kept non-toxic until they are required. So we know that the binding or the association with peptide 2 inhibits toxicity. And so this could be a way of how the fungus actually protects itself, because it's processed in the Golgi within the Candida cell. And so that might be a mechanism. But we are not 100% sure whether the membranes of Candida albicans are susceptible per se. And finally, the third hypothesis, and that's my favorite one, but difficult to find out, it might be that the other peptides are transported into the host cell via the activity of Candida lysine. So Candida lysine could cause pore damage, and the other peptides are released and transported into the host cell. And here they would have effector peptide functions. So that's my favorite idea. I don't have any evidence for this, but it would completely make sense. And if you're aware of plant pathogenic fungi and bacteria, they have hundreds and hundreds of effector proteins and effector peptides. Bacteria use type 3 secretion systems or type 6 secretion system to inject proteins into the host cell, and these modulate the host cell to their favor. Plant pathogenic fungi have the same, but there's not a single effector peptide of protein found in human pathogenic fungi. So this is my proposal we are working on.
Right. So Amparo, maybe you want to ask? I can hear you. Amparo, write the question because we can hear you.
There's one in the chat, maybe.
Yeah. So is Candida lysine or a similar protein present in other fungi?
Yep, very good question as well. Interestingly, there is a similar protein in Candida tropicalis and Candida dublinensis. Also, these two are less aggressive as Candida albicans. In particular, Candida dublinensis is very similar to Candida albicans, but clearly less pathogenic. And if you look at the synthetic peptides, which are homologous to peptide 3, so Candida lysine, then these synthetic peptides are actually more aggressive as Candida lysine itself. And that is, of course, strange, but on the other hand, these two other species do produce far less hyphae, and we don't know exactly how this is regulated, so that suggests that there's potentially other functions which we are not aware of. But basically the answer is there are a few, and we don't know exactly what they are doing. This is work from Julian Naglick's team.
Hi, Bernhard.
Hi.
I am Amparo. How are you?
Yeah, I saw you.
I decided to use the Roberto microphone.
Right. Yeah.
I want to question, my question is about the other Candida. You have a strong work with Candida albicans and also with Candida glabrata, but you know that I am working in a food research institute, and you know well that we are interested also in food yeast that also can produce infections. In this case, it's an other not very well known Candida species like Candida valida, Candida cruensis. Do you have information, or do you think that the mechanism can be similar to produce the pseudofilamentation and perform the penetration inside the cells to produce the damages? Or what do you think about the other species? You have some ideas? Or,
yeah.
Because this is very interesting for us.
Yeah. Okay. What I don't have much experience with the other Candida species, but we know pretty well about the top four: Candida albicans, Candida tropicalis, Candida parapsilosis, and Candida glabrata. As I said in the beginning, glabrata and the three others, they are not related at all, but they're all pathogenic. Candida glabrata is unable to produce hyphae, yes, and it's still pathogenic. So there must be very different mechanisms. And I would say that Candida albicans is actually an exception, but it might be, and in particular for the morphology, because it's a very efficient producer of hyphae formation. And this might explain why Candida albicans is by far the most aggressive one from these top four. However, I should also mention that depending on the clinical isolate, you might also have some which are less hyphae producer than the one which we are working with. We are working with strain SC5314, and that is in fact one of the most aggressive ones we can get, which is good for us because we can work with this, we can elucidate all the functions of hyphae, but this is more or less an exception. And the explanation for this is possibly that hyphae formation is not only an advantage for this fungus, it's also a disadvantage. And there's interesting work published recently about the colonizing form, the commensal form. The commensal form of Candida albicans seems to be yeast cells. So somehow hyphae formation in the gut is detrimental, potentially because these hyphae are too aggressive, they invade, they cause a protective immune response, and then they are cleared. So as a commensal, the fungus has to be quiet. And maybe that's a success of the others, because glabrata and parapsilosis almost cause no damage of epithelial cells if you do this in vitro. So yeah, hyphae formation is clearly key for at least Candida albicans.
Thank you, Roberto. Thank you very much. See you later.
See ya.
So yeah, thank you, Bernhard. I had another question. So I wanted to ask you about the limitations in the growth of yeast inside the human body. So you talk about zinc or iron, but also nitrogen. What do you think is the most important, not for the pathogenicity mechanism, but just for normal growth of the yeast? What do you think is more limiting?
Well, I can't judge what is most important, but I agree with you. It's not only the metals which are required, but it's also all other types of nutrients. I can say something about nitrogen, because nitrogen is of course required as well. Because I've worked for a long time on proteases. There's an entire family of secreted aspartic proteases of Candida albicans, and tropicalis, parapsilosis also have these proteases. Glabrata has no secreted proteases, but cell surface associated proteases. So obviously the fungus, I mean, there must be a reason why Candida albicans has 10 different proteases of this type. And I guess this is nutrient acquisition. I guess maybe it's similar to what I have shown with the damage at this hyphen tip, that there's protease activity digesting host proteins to get nitrogen from the host.
Okay, so it's not to break down the proteins that are like making the defense. It's just for the nitrogen acquisition.
No, I wouldn't say it's either. I mean, I would say it's both.
Both.
Maybe one principle of what I have learned in my career that is, if you have a protein, it rarely has only one function. It's most of the proteins which we are looking at, they have several functions. And proteases are well known. It was a very hot topic about 15, 20 years ago. Well known to digest immune proteins, and factor H can be, complement factors can be digested. Immune globulins can be digested. So I guess it's both. I guess it's also degradation of as immune defense mechanism, but getting nutrients as well.
Okay, thank you.
By the way, we're also working with lipases. Another family of 10 different lipases
okay.
also suggests that there's activity to digest fat into fatty acids, and they might be used both for nutrients but also for competition with other microbes, because fatty acids can be inhibitory, so we are working on this.
Okay. So yeah, Amparo also, she's working at the EFSA, and now they are discussing about the security of yeast. So she's asking about if you have to define a yeast is safe or not, which mechanisms will be most adequate?
Difficult to say.
What do you look for when you have to define a virulent yeast?
We discuss this also in a related context, because the idea is to have probiotic, antagonistic activities. And we are working with lactobacilli, and lactobacilli are in use as probiotics, and we are investigating how it actually is inhibiting, in our case, the fungus. If you talk to clinicians, they say, oh, this is critical, because even these non-pathogenic microbes might cause disease in these highly susceptible patients, which actually get these fungal infections. So potentially we could find out the protective mechanisms of these probiotics and use these mechanisms. For example, let's say if they produce fatty acids, which are inhibiting Candida albicans, for example, then potentially we can use the fatty acid, because this is not infectious. So maybe we have to elucidate the mechanisms and can use these mechanisms to develop therapies without using living probiotics.
Okay. Yeah. So, yeah, and there's any other questions? If not, we don't want to steal more time of your valuable time. And yeah, for me, it's really, it has been a pleasure to have you here. And I hope we can see each other in some meetings ahead of us. And I think everybody is thank you for you to come here to talk about this interesting topic. And yeah, I don't know if we cannot make applause, but if they will be here, everybody is writing in the chat.
Yeah, I see the nice chat. I would like to thank you as well for inviting me, and hopefully next time I can be in person in Valencia.
Of course.
Be there in person and maybe having a nice dinner afterwards, because I love Spanish food.
You can take advantage of our climate and food. Maybe next year. It will be really a pleasure to have you. And yeah, I have no more words than thanks. And yeah, we hope to see you soon. And thanks for your talk.
Yeah, thanks to you and thanks to everybody listening.
Bye.
Thank you very much. Goodbye.
Bye.